Today, my paper title is Function of Medicinal Flora in Human Health Disorder. Today, we are talking about this point abstract introduction, review literature, methodology, review uh, result and discussions and conclusion. We have collected 20 plant species from 19 genera and 15 family were documented, whereas herbaceous plant species have been dominant in the study area. Among these 20 plant species, shrubs have 15% and timber have 20% and climber of 15%. Euphorbiaceae become the leading circles of relative amongst acute medicinal plant species. Out of 20 plant species, leaf is used of 6 plant followed by root, stem and fruit, and whole plant of 2 species used as ethnomedicinal plant from the remedy of various elements. Present examine may be useful to ecologists and taxonomists and preserve the culture identity of human besides the providing possibilities of reputations, advertising, control and protection of indigenous understanding of a community on a medicinal plants.
Hello, hello, Dr. Jitendra Singhediya, sir.
Is I audible, sir?
Yes, yes, very much audible, very much audible.
Okay.
Please continue.
Introduction. So we are talking about the medicinal flora, which is a healthy source of additives and another things. An element from these floras play a crucial function inside the improvement of human culture throughout the complete international. Plant is a crucial supply of medicine and perform a key role in global health. And plants are crucial to the functioning of all social societies and the operational all ecosystem. 15,000 plants are used for medicinal purpose It is the reported by given in reference.
So you are not audible.
The Narmada Valley is divided, flowing in this area. Sindhwa is first become constructed in 10th century, and it have a four-dimensional gate fort situated in the middle of the town. It is a very good example of four-dimensional gate, and elevation is 409 meters, and literacy rate is 63%. High amount of exotic floristic element migrate in Indian flora. Vegetation of Sindhwa is classified by champion and shade, and there is a small painting on finishing on these locations on some of the literatures available, which is shown in last portion of the research paper. Now we are talking about the material and method. A systematic plant survey and collection was accomplished on 2021 to 2022 by well plant schedule. A plant species occurred in one kind of seasons. All habitat of various ecological niches of the study area were surveyed carefully. Complete of 100 local inhabitants was interviewed through questionnaire methods. Identifications obtained by quantitative analysis. Field observation and field data were noted down in field notebook. Collected specimens were kept in between folding of blotting papers, pressed in the field press and dried. Dried plant specimens were preserved by dipping of whole saturated solutions, 2% saturated solution of mercury chloride and isolate alcohol. This is the photograph doing collection and preservation and making herbarium seed and identification in Botanical Survey of India, which is situated in Allahabad. And this is the vegetations of our area. This is the deciduous forest type of vegetation, and the Diosporus melazalan forest is also situated in some part. And dry deciduous forest is have looking in this picture, and Tectona forest is also available in our area. And plant series is done by standard approach by Jain and Rao. Plant specimen preserved by dipping the whole specimens in saturated solution mercury chloride and alcohol and making herbarium seed and deposit into government PG college Sendwa. And some plant have identified by floras like hooker, gamble, hence in our state flora I have also included which is written by Verma and his team, and Mudgal and Dadichi and Shah have also consulted the surrounding states taxonomic literature. Some plants they have which is difficult to identify, which is identified in Botanical Survey of India. Now we are talking about the results of our study. The medicinal flowers play a vital role in element prevention, and their marketing and use fit into all the current prevention techniques. 20 plant species belonging to the 19 genera and 15 families were documented into modern day study. Those medicinal plant species are used generally as an ethnomedicinal against diverse diseases such as a cough, fever, allergies, and diarrhea, and many other diseases. Some plant dispensed as a herbal 10 plants, 3 climbers, and OD plants, which is called a timber of 4 plant species used in the different diseases. This is the our main result, which is shown in table. The first of botanical names and second is the family and its life form also shows harbor, shrub, tree, climber, etc. And plant part is used. It is shown in table and preparations of medicines form are given in this column. And the last column is shows the disease like Dioscorea bulbifera plant is belonging to the Dioscorea family, which is habits is herb, and plant part is used of root, and it's a decoction made for the disease of the abdominal plant. In another plant have also second plant Embolica officinalis, which is called aula. Its fruit is eaten in anemia, and Euphorbia hita is this plant stem part used for making decoction and given in asthma, and Diplopsychus palmatus is also given in constipation, and Senatora is used for cough. Symbopogon martini is the belonging to the Poaceae family. It is the grass and herbaceous plant. Its leaves is used for making a juice, and juice is given in diarrhea disease. Icomia fistula is also used in fever, and Cuscuta is jaundice, and Balanitis egyptica is used for malaria, and another plant is shown in next slide. Eucalypta prostata is also called Bhrangaraj. It is belonging to the Asteraceae family, and his whole plant is used for skin disease. And Dhatura metal is used in also skin disease. Cissus quadrangularis plant is applied for snake bite. Curcuma aromatica, it is also called jungli haldi, which belong to the Gingiveraceae family, and its habitat is herbaceous plant. It is root part is used for making a decoction and used for tonic. Jatropha carcass is also used in ulcer, and Ficus religiosa plant is used for ulcer, and Cynodon dactylon, it is also member of the Poaceae family, and it is make a decoction and given in urinary infection, and Ficus bengalensis is also used for vomiting. Now we are talking about category-wise distribution of the taxa. 3 plants out of 12 is climber, and 10 plant is herb, and 3 plant are shrubs, and 4 plant are trees. Herbaceous plant species were the dominant with 50% among the vegetation studies observed with the aid of the shrub, tree, and climbers. Euphorbiaceae is the main dominant family on the observer family. Plant component made the use of classes in the table shown. The 2 plant used as a whole plant, and 4 plant is used as a root, and 5 plant species of stem is used for different diseases, and 6 plant leaves are used in different diseases, and these are the table shows. Now it is the geographical design of the result. We are talking about the conclusions of our research article. First of all, uses of natural medicinal drug in the treatment of disease will grow every day. The natural and precise medicinal flowers are used for curing these diverse element and profits technology. Ayurveda and different Indian literature have referred to the use of vegetation in remedy of various human elements. The cutting edges observer revealed that the significance to record and launch a listing of all viable flowers which can be used in ethnomedicinal practice in the examiner area. Medicinal flora is vital delivered to fight the acute illness in all over the worldwide. Dynasty antimicrobial, antiviral, and pharmacological studies are required the efficiency and protection of the medicinal flora species. Human activity has affected the floristic compositions of the local. Existing study useful for the further researchers who have worked on the ethnomedicinal vegetations on this place. These are the some references given in literature. It is also, and now we have thank you for listening me.
I hope Dr. Jitendra Sinkedia sir, you complete your presentation.
Yes, sir.
Okay. Thank you so much. Now, the next invite delegates is Kunwar Sarvender, Department of Botany, Kusum University, DSB Campus, Department of Botany, Nainital, Uttarakhand. Dr. Kunwar Sarvender. Dr. Kunwar Sarvender. Dr. Kunwar Sarvender. Okay, next one. Next participant is Ajay Kumar Gautam, Sivli National College, Azamgarh. Next participant Ajay Kumar Gautam, Sivli College, National College, Azamgarh.
Mr. Siddharth,
Yes, sir.
may,
Yes,
may, I,
sir.
may I audible?
Yes, yes, yes. Very much audible.
Okay, thank you.
Please share your screen.
Okay. My screen visible?
Yes.
Okay.
No, 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 no.
No visible?
Yes, now visible. Very much.
Okay. I, Ajay Kumar Gautam, Research Scholar, Department of Botany, Sri National College. My topic is impact of anthropogenic activities on freshwater lake, Sirantal. Now come to the introduction. Water is the most valuable resources on this planet. Water is essential for our life. It is necessary for the growth and preservation of every aspect of society dynamics. Due to its unique physical and biochemical features, it is a critical component of cells, accounting for 70 to 90% of the total mass of each cell. Further, due to hydrogen bonding, water molecules exhibit properties of cohesion, subsistence, solvent capability, capillarity, and moderation of temperature. In addition, since it is a liquid, it serves as a medium for the dissolution and interaction of other substances, including polar and ion molecules, that account for the bulk of biochemical reaction in living organisms. Surface water bodies have long been a vital resource for meeting a wide range of human needs, but owing to a variety of anthropogenic activity, the water quality of such resources is now under significant environmental stress, and the situation is much worse in lentic water body. There are several religious activities that are responsible for the contamination of the lake due to immersion of detergents as soap, oils, floor, moss, bathing are also one of the significant reasons that degrade the water quality of the lentic water body. Floral offerings, adding acids, floor, urination, and bathing could raise the nitrate and chloride content of water in accordance with pilgrims' high organic matter and pollution load on water bodies. According to WHO 1993, water is responsible for about 80% of all human illnesses. As a result, continuous monitoring of such lentic water body is critical for physicochemical examination to determine the appropriateness of the water in use, not only to avoid disease outbreak, but also to prevent future degradation of water. The current study was conducted on Siravantal to study 12 physicochemical parameters or objectives, assessing water quality and suitability for drinking and other purposes, analyzing annual and seasonal variation of selected physicochemical parameters in water, determine the various physical chemical characteristics of water quality parameters present in the lentic water bodies, assess to sources of pollution. Study area. The study area is Siravantal, which lies to the south of Adamgarh district with geographical coordinates of 25.5 to 20 degree latitude and 82.5940 degree longitude. It is located near Adamgarh Varanasi Road in Siravan village. The area of the lake is approximately 21.41 hectares. The lake is mainly used for fishing, watering, and recreation. This is the image of Google photograph of Sri Ramantal. Material and method. The current study examined 12 physicochemical parameters, pH, ES, EC, TDS, TSS, TS parameters were determined by standard procedure AWWA, American Water Work Association, and AFA 1999. DO, dissolved oxygen by Winkler method, total chloride by Argento method, nitrate, nitrogen, and ammonical nitrogen by salicylate method, CSIR 1974. And orthophosphate phosphorus as well as total phosphorus by stannous chloride method. Now come to result and discussion. In the current study, the pH value varies from 7.4 to 8.6, indicating that water is lightly alkaline in nature. The increasing value of pH is related to rising pressure of pollution due to human activity. The EC value in this lake water sample 152 to 365 microsiemens per centimeter, showing that the EC values were greater than recommended limit, nearly limit. Uh, Dissolved oxygen varies from 4.5 to 7.7 milligram per liter. The value of oxygen maximum at site 1. The BUD value is 80 to 26 milligram per liter. The BUD value is not accepted for drinking purposes. The value of TDS, total dissolved solid, 74 to 165 milligram per liter. Total chloride concentration range from 27 to 36 milligram per liter. A high chloride content shows a considerable level of organic contamination. This is the graphical representation of the result in discussion. And now come to the conclusion. The current research provides a better insight into the lake quality. The physicochemical analysis indicates a rise in the parameters of water quality. After examination, it is clear that the primary causes for the degradation of the water quality of the lake are eutrophy due to the use of pesticides and fertilizers. The direct discharge of sewage from surrounding village increases the nutrient. This causes many harmful effects on the aquatic fauna and flora. As a result, there must be appropriate disposal and management of wastage. An action was developed for the conservation of these lentic water bodies. Now, acknowledgement, I wish to express my profound gratitude and sincere thanks to Research Guide Dr. Abdullah, Associate Professor, Department of Botany, Civil National College, Azamgarh, for valuable guidance, constant encouragement, and untiring help, which had been a source of inspiration at all stages of the work, made me to successful in the completing study. Thank you so much. Mr. Siddharth,
Ji, thank you so much, sir.
any query?
This is your own research work.
Yeah. I
Uh,
selected eight lentic water bodies of Adamgarh Mau district. One of them, that is Ramanta.
okay. So this work was performed at where?
Yeah.
This work is performed at where? मतलब in your lab or
I,
any?
some parameters in Civil National College laboratory and some parameters from private laboratory because there are
Okay.
some
Okay.
infrastructural issues.
Okay. Thank you so much, Dr. Ajay Kumar Gautam, sir.
Thank you so
Thank
much,
you.
Mr. Siddharth. Thank you.
Next, I would like to invite our next invite delegate Seema Kumari, Career Point University, Department of Chemistry, Hamirpur. Seema Kumari, Career Point University.
Excuse me, sir. I'm audible, sir, now?
Yes. You are very much audible.
Okay, sir. Okay, sir. So, sir, can I share my screen now?
Yeah, sure.
Okay, sir. Sir, my screen is visible now.
No. Your skin is not visible.
Okay, sir. Okay, sir. Sir, I have to go open share tray.
Yes, open.
Share tray, sir.
Yes.
Then, sir, share
Presenter
screen.
mode. Share content just नीचे presenter mode. After that, you see the screen. Just click the
Hanji,
screen.
okay, sir. Sir, now here is. Sir, I don't see the presenter mode here. Sir, I have to open share tray. Then, sir.
After share, you can just click the screen button.
Uh,
You see a two button, screen and window. You just click the screen button.
Screen and window. I have to see the, sir, entire screen, sir.
Yeah. Entire
Oh,
screen.
sir, entire screen is okay, but sir, I can't got the option to share it entire screen. So now, open share screen, screen share. Sir, I have to, sir, click on the entire screen or window, sir.
Entire screen.
Sir, entire screen is already okay, but why?
You just share me your PPT
Yes,
and
sir.
please wait for your next after next.
Sir,
You just
how
share
can
me
sir, how can I share my PPT with you?
by mail ID ace.research.edu@gmail.com.
Okay, sir. Sir, I will. I will screen share. Sir. Sir, now they are visible or not, sir?
No, not visible.
Not visible, sir.
Any other organizer can see her slides?
No.
Okay.
I am unable to see it. Sir, I can join from the mobile phone, sir.
Yeah, you can, but then you can share your PPT.
हाँ जी सर.
Yeah.
सर प्लीज मैं अभी सर आपके साथ शेयर करती हूँ अपनी पीपीटी.
Yeah, sure. Karisma ma'am, please contact her. And next one is Jogender Prasad Pandey, Department of Zoology, Government Model Science College.
Sir, please, will you please give share your email ID with me, sir?
Please contact Karisma ma'am. Please contact Karisma ma'am and mute yourself. Then I will call you next. Jogender Prasad Pandey. Next one, Alekhya Sarkar. Next one, Alekhya Sarkar. Kumar Sarvinder. Kumar Sarvinder.
Hello.
Yes.
Kumar Sarvendra has shared his PPT with me. I'm just sharing. Wait.
Excuse me, sir.
Yes.
Sir, myself Seema, sir.
Yes, yes, please say.
Sir, now, sir, my slides are visible.
Yes, your slide is visible.
So, sir,
Please proceed. Please proceed.
Okay, okay. Thank you, sir. Thank you, sir. Good afternoon to all of you. Myself, Seema Kumari, PhD research scholar in the subject of chemistry from Career Point University, Hamirpur, Himachal Pradesh. Today, I am here to deliver a presentation on effect of divalent metal ions doping on structural properties of nickel ferrites. The main outlines of my presentation are introduction, methodology, characterization, results, and then conclusion. First of all, what do we mean by the term nanotechnology? The nanotechnology is that technology which deals with those atoms and molecules whose dimensions lies in the nanoscale, that is from 1 to 100 nanometers. The words of Richard Feynman, there is plenty of room at the bottom, in a meeting of California Institute of Technology on 29th December 1959, gave birth to nanotechnology. The particles whose size lies in the range of nanoscale are known as the nanoparticles. The large surface area and the high surface energy of these particles provide them the unique characteristics. And due to these unique characteristics, these particles have wide applications in various fields. In recent years, among these nanoparticles, the magnetic materials have been the focus of much research. Because of their attractive properties, they are widely used in the field of biomedicine, magnetic resonance imaging, data storage, environmental remediation, magnetic cooling, etc. So now why these particles have great demand? It was due to their tracking as well as manipulation under the influence of applied magnetic field. So finite size and surface effects of these particles increase their utility. Among magnetic nanoparticles, the ferrites are of great interest. Ferrite is a ceramic material made by mixing and firing large proportions of ferric oxide along with the small proportion of one or more additional metallic elements. And in ferrites, the ferrites belong to the category of ferrimagnetic material. That is, they can be magnetized or attracted by the magnet. So based upon the resistance to being demagnetized, ferrites can be further classified into two classes, and they are the soft ferrite and hard ferrite. Soft ferrites have low coercivity, so they can easily change their magnetization, while the hard ferrites have large value of coercivity and less susceptible to demagnetization. These ferrites can be used to make the permanent magnets, while the soft ferrites can use to make the temporarily magnets. Among these ferrites, based upon their structures, they can be further classified into three classes. They are the spinal, hexagonal, and the garnet ferrites. The spinal ferrites have the face-centered cubic structure. Hexagonal have hexagonal structure, while the garnet ferrites have the structure of silicate minerals. In the spinal structure ferrites, they have the general formula Ab2O4, where A belongs to a divalent metal ion category and B belongs to the trivalent metal ion category, and specifically the ferric ions. In the spinal structure ferrites, the divalent and the trivalent metal ions, they are coordinated tetrahedrally as well as the octahedrally with the oxygen anions. As the structure shows here, the spinal ferrites has 64 octahedral sites and the 32 tetrahedral sites. The number of octahedral sites are twice than the number of the tetrahedral sites. Now, based upon the occupancy of these divalent and trivalent metal ions in the tetrahedral and the octahedral sites, the spinal ferrites are further classified into mainly two classes. They are the normal spinal ferrites and the inverse spinal ferrites. In the normal spines, as shown in the structure figure A, the divalent ion resides in the tetrahedral sites, while the trivalent ion resides in the octahedral sites. And totally in a unit cell, there are 8 tetrahedral sites which are occupied, and the 16 are the octahedral sites which are occupied by the ions. So therefore, in a ferrite unit cell, there are 8 divalent ions and the 16 trivalent ions, and as well as the 32 oxygen ions are there. On the similar trend, in inverse spinals, the divalent cations resides in the octahedral sites, while the trivalent ions, they resides equally in tetrahedral as well as the octahedral sites. Now, among the spinal ferrites with nickel as a divalent cation are referred as the nickel ferrites. Nickel ferrite possesses the inverse spinal structure in which the 8 tetrahedral sites are occupied by the ferric ion and the 16 octahedral sites, they are occupied by the ferric ion and the nickel ions in equal proportions. Now, in order to improve the properties of nickel ferrite nanoparticles, it might be doped with the other divalent metal ions. In my present study, I doped nickel ferrite with the magnesium and the calcium ions. They belong to the category of alkaline earth metals. And modification in structural, electrical, optical, as well as magnetic properties of pure ferrites can be achieved by the divalent metal ion substitution. And these properties can be tailored by the choice of synthesis route, composition of chemicals, nature of dopant ion, as well as the thermal conditions. Methodology. During the synthesis, there are the two main approaches which can be applied to synthesize the nanomaterial. They are the top-down approach and the bottom-up approach. In the top-down approach involves the breaking down of bulk material into the nanostructures material, while in the bottom-up approach, the molecular components arrange themselves into more complex assemblies, atom by atom, molecule by molecule, or cluster by cluster from the bottoms. So next one is the synthesis methods which are adopted in the bottom-up and the top-up approach are, during the bottom-up approach, they are atomic or molecular condensation, vapor deposition, sol gel, spray pyrolysis, co-precipitation, and bioreduction. During the top-down approach, the method adopted are sputtering, chemical etching, thermal or laser ablation, mechanical wall milling, explosion process. During my studies, I adopted the co-precipitation method, which belongs to the category of bottom-up approach. So during the synthesis, there is a mixing of ferric salt, nickel salt, as well as the divalent metal ion salts in a stoichiometric amount. That is in the ratio of 1:2. They are firstly dissolved in the distilled water, stir on a magnetic stir for a particular time, and then heated the contents at a desired temperatures. Then added the sodium hydroxide, which will acting as a precipitating agent, drop-bys in order to adjust the prescribed pH of the solution. Again heated the contents for a particular time, and then there is a generation of precipitates. After that, the washing, drying, and grinding of these precipitate will be there. And finally, the grinded sample will be calcinated at a desired temperature. And finally, we will got the nanoparticles of nickel ferrite as well as the divalent metal ion doped nickel ferrites. During the nickel ferrite synthesis, we don't take the metal salts. Now, characterization. The synthesized samples can be characterized by various techniques, likewise X-ray diffraction, scanning electron microscope, transmission electron microscope, thermogravimetric analysis, vibrating sample magnetometer, Fourier transformation infrared spectroscopy, ultraviolet visible spectroscopy, energy dispersed X-rays diffraction. Regarding the results that I have got presently are only the XRD and FP-SAM result. The results of other characterizations are still awaited. The obtained XRD spectra within the range of 20 to 80 degree of nickel ferrite and magnesium and calcium doped nickel ferrite is as shown in the figure. The sharp peaks corresponding to crystal planes of 220, 311, 222, 400, 511, 440, 533. And the presence of these peaks along with the diffraction from these crystal plane shows that the resulting structure is face-centered cubic. And then the structure parameter can be calculated. The crystalline size can be calculated by using the Debye-Satcher formula, which is D is equal to K lambda upon beta cos theta. Then lattice parameter A is equal to DHKL, square root of H square, K square, and L square. Volume is equal to the cube of lattice parameter, and X-ray density is equal to Zm divided by N naught into AQ, where Z is the number of atoms per unit cell, M is the molecular weight of the sample, N naught is the Avogadro's number, and AQ is the volume of the unit cell. DHKL is the interplanar spacing, and HKL are the Miller indices. Lambda is the wavelength, beta is the full width half maxima, and theta is the diffraction angle, and K is the Satcher constant. And then the obtained results are, there is a in the results we found that there is decrease in the crystalline size on doping of alkaline earth metal to the nickel ferrites, decrease in size from 56.4 nanometer to 50.2 nanometer, decrease in the lattice parameter, then decrease in the unit cell volume, as well as the decrease in the X-rays densities were found in the obtained results. The resulted FSM micrographs clearly indicate there is a decrease in the crystalline size on doping of magnesium and calcium to the nickel ferrites. The resultant micrograph also shows that the particles are spherical in shape, uniformly distributed, as well as get agglomerated. Agglomeration occurs due to the dipolar interaction among the particles. There are so many characteristics of nickel ferrites, and just like small size, high surface area, magnetic separation, recyclability, requirement of small amount, simple preparation, mechanical hardness, high Curie temperature, thermal stability, easily recovered and reused, as well as the chemical stability. So they can be used in so many fields, just like as a photocatalyst, in water purification, biomedical field, antimicrobial agent, sensors, adsorbent, microwave devices, electronic devices, etc. Lastly, the conclusion. Nickel ferrite and alkaline earth metals doped nickel ferrite were synthesized successfully by the co-precipitation method, and doping shows there is decrease in the crystalline size. FSM analysis also shows the spherical shape, agglomeration, and the presence of pores on the surface of nanoparticles. Finally, there are the challenges during the use of these magnetic nanoparticles. And the main challenge is of the long-term use of magnetic nanoparticles is biocompatibility as well as toxicity. So to overcome these challenges, critical and constructive research are demanded to design and fabricate magnetic nanoparticles for their applications in different fields. Thank you.
Thank you so much, ma'am. It's very nice presentation. Can you show me your XRD graph slide?
Yes, sir. Yes, sir. Oh, sorry, sir, XRD.
Yes. Ma'am, how to calculate the plane? This is 221, 311, or 400.
Sir,
From
that is from
literature
the
or is
literature.
calculated by numerical any?
No, sir, from the literature, sir.
Okay, and the next third slide, that is 19th number slide.
19th, sir.
Yes.
Yes, sir.
How to calculate these values from
Sir,
XRD graph?
yes, sir. From XRD graph, the crystalline size will be calculated by Dweyer-Sucher formula, sir, which is D, where D representing the crystalline size in nanometer. K is the
From the graph, what is the value of cos theta?
sir cos theta value will be given in the sir graphs. That will be just like sir, just sir from here. That is a diffraction angle, sir.
Yes. So what is the value of cos theta for the plane 222?
Sir, from here, the value is sir around about 27, sir.
It is not a sin theta value or a cos theta value?
Sir, it might be the cos theta. Sir, it is a theta value. Then, sir, this is a value in degrees, sir. We have to convert it in the radians, sir.
Okay.
Yes, sir.
And HKL value?
Sir, they are the Miller indices, sir.
Okay, from literature?
Yes, sir. From literature, sir, we will match the angles with the corresponding literature. Then, sir, assign the values of HKL. That is a Miller indices.
Okay, okay. Thank you so much, ma'am. Thank you so much.
Oh, okay, sir. Thank you, sir.
Thank you. Now I would like to invite our next invite delegates, Dr. Biswajit Das.
Am I audible?
Yes, yes, very much audible.
Okay. Thank you, sir. Is it visible, sir?
No, 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 not visible. Yes, very much visible. Please make a full slide, full screen.
Is it okay, sir?
Yeah, is it okay? Please make a full screen.
Now is it okay?
Yes, now okay.
So thank you very much, sir and madam, for inviting me in this sixth annual meeting of IJET and third international conference on recent trends in life science and biotechnology. So today I'll be presenting my published review article, that is anti-cancer stem cells potentiality of an anti-malarial agent quinacrine and old wine in a new bottle. Basically, quinacrine is a multipotent drug. Now, scientists found that a very interesting property of quinacrine in the field of anti-cancer research. So this is a plant-based product and derived from cinchona bark of cinchona sp. And initially, it was used to treat for amoebasis, lambiasis, giardiasis, and other diseases. And now this drug is under clinical stage trial 2 in prostate cancer. And the interesting fact about this drug is this is highly effective against cancer stem cells. We know the major challenge regarding the anti-cancer therapy is the cancer stem cells, because easily we can kill cancer cells, but when it comes to cancer stem cells, there there is no easy process or easy way to kill cancer stem cells because of many reasons, mainly their high self renewal capability, DNA repair capability, and multi-drug resistance properties. So previous studies, and we have published many papers regarding this anti-cancer stem cells potentiality of quinacrine. And recently, we have generated quinacrine gold hybrid nanoparticle. We combine this drug with nanoparticles of silver and gold, and the effectiveness of this drug far better from the normal case. So in this way, we are trying to solve or at least give some relief to cancer patient and cancer therapies. So in this way, you are trying, and here another strategy also due to the to increase the bioavailability and effectiveness, the liposome encapsulated QC has been used in cancer research, which can selectively target cancer cells and cancer stem cells. So this is the main schematic diagram. So here we can see that in cancer cell population, there are cancer cells and cancer stem cells also. Quinacrine is effective in the killing of cancer cells as well as cancer stem cells. So in case of cancer cells, it increases the apoptosis and inhibits cell growth, inhibits topoisomerase activity, induce autophagy, and inhibits the DNA repair mechanism. And in case of cancer stem cells, as we know, cancer stem cells are mainly responsible for angiogenesis, metastasis, and this drug also able to downregulate the CSC signaling cascade. And when we are
Biswajit sir,
applying this drug,
excuse me Biswajit sir, एक्चुअली आपकी जो स्लाइड्स है वो फ्रीज हो रखा है.
sorry.
Your slides is in freeze mode. Yeah. Now visible. Yes, carry on, please.
Yeah. So two types of therapies like quinacrine able to kill cancer cell as well as cancer stem cells, and we know the angiogenesis metastasis are the main properties of cancer stem cells. So here this drug is potentially killing this cells, means bulk of cancer cells by various mechanism, what I already told, but the time slide was not there, so again I'm saying. And apoptosis, it induce apoptosis, reduce the cell growth, and decrease the topoisomerase activity, induce the autophagy, and deregulate the DNA repair mechanism. Ultimately, these are leading to cell death, that is cancer cell death. Next, what are the anti-cancer potential of quinacrine? So I have enlisted out this. First one is DNA intercalation, topoisomerase activity on DNA and RNA polymerase activity, cell growth and cell cycle, autophagy, apoptosis mediated cell death. So in all these important properties of cancer cells, quinacrine is effectively deregulating this mechanism. So here I explain in detail, but as we have very short time, so I'll not be able to explain everything in detail. But here you can see the QC role of QC in DNA intercalation, topoisomerase activity. It inhibits the topoisomerase activity, and it also inhibits the DNA and RNA polymerase activity, and it also gives some significant effect by inhibiting cell growth and stalling the cell cycle. Next, as I already told you, able to induce the autophagy and apoptosis related cell death. So in case of apoptosis, the researcher found that it induced P53 upregulation, activating VAX and changing the ratio of VAX BCL, and ultimately leading to apoptosis cell death. And this drug also downregulates the apoptosis protein 1, that is CIAP1, and upregulates the VAX. So next, I have schematically represented the effect of quinacrine on cancer-related cell signal. There are many types of cell signaling where QC is playing a crucial role, like in case of PI3K-AKT-mTOR pathway, trail DR5 pathway, NF-kappa pathway, and some other signaling like STAT and FACT trapping. And in this way, QC is deregulating the cancer-related cell signal. It inhibits the AKT phosphorylation at particular serine 473, and it also induces the death signaling like the death receptor DR5. So in this way, this drug also targeting cancer-related cell signaling. And next one is, as I told, whenever we are putting this drug, combining with other drug, like their synergistic effects of anti-cancer therapy have been found. In my published article, there are lots of drugs, but as the space also very limited, so I select 3 or 4 drugs here. And that is APT-888, 5-fluorouracils, paclitaxel, and sedinarine. These are and well-known drug, and already these are showing the very significant result whenever we are applying these drugs with in combination with quinacrine. So, as I told that quinacrine is having very interesting property, that is, it specifically targets cancer stem cell. The main reason behind cancer relapse are cancer stem cells. So, it is very important to target those root cause of cancer. That is in the my several methods like liposome mediated delivery of quinacrine for targeting CSC as well as cancer cells. Metastatic quinacrine has a metastatic inhibitory action. As I told, the metastasis is regulated by cancer stem cells, and it also have anti-angiogenic action. It is also very important properties of cancer stem cells. And next, QC helps to inhibit DNA repair activity in metastatic breast cancer cells, and applying a novel form of QC. What I told already, that nanoquinacrine and quinacrine hybrid gold and silver nanoparticle. So these are the mechanisms I explained in detail. Yeah, here is the some of the signaling, that is stem cell signaling, Hedgehog signaling, WNT beta catenin, notch, nectin-4. QC different forms means now newly synthesized nanoquinacrine and gold hybrid nanoparticle. They are found to inhibit this uh, cancer stem cell signaling via various mechanism by inhibiting the cluster of GSK3 beta, GLI1 and beta catenin, and by inhibiting the nuclear translocation of nectin-4, which is well known for cancer angiogenesis. So in this way, quinacrine is deregulating this signal, and ultimately it inhibits the cell growth and development, cell proliferation, and base accession repair activity. So coming to the conclusion, the QC is known to be a safe drug. As I told, it is a plant-based drug with a minimum side effects, and this drug played a vital role in cancer treatment. And studies are still going in order to increase the bioavailability and effectiveness. And this drug is why so special, because this drug not only targets cancer cells but also targets cancer stem cells. Such potentiality of this drug will surely add benefits to the ongoing cancer research. And it was found to be effective in treating cancer when directly injected inside the tumor. And this drug can target cancer stem cells when administered with other drugs through liposome, as I already mentioned. So in modern days, modern techniques also coming, some new forms of quinacrine, that is nanoquinacrine and hybrid nanoparticle, have evolved to target cancer stem cells, cancer cells, and more specifically to cancer stem cells. And these new forms of QC exert their effects by deregulating stem cell related signaling pathways such as WNT, notch, nectin-4, and after all this, QC has also the potentiality to inhibit metastasis and angiogenesis. These are two major hallmarks of cancer progression, which are also main properties of cancer stem cells. Conclusively, we can say that an antimalarial drug, quinacrine, has been rediscovered as an anti-cancer drug, showing anti-cancer potentiality when administered alone or in combination with other drugs. Also in its novel. So, the old saying, old wine in a new bottle is apt for suitable for this drug. And in case of future research, we are expecting that QC's ability to heal CSC and will surely open the door of various applications in cancer research and how it is killing the cancer stem cell in the bulk of tumor cells. That is also research topic for future. Thank you very much. If you have a question, I have provided my email ID also. You can ask me.
Thank you so much, sir. Very nice presentation. It is very fruitful and very informative talk. Anyone want to any inquiry? Anyone want to any questions? Raise your hand. Okay, thank you, sir. Thank you so much.
Thank you very much, sir.
Now the next invite delegates is Dr. Nanu Safakatullah, Department of Bioscience, Mangalore University, Karnataka. Dr. Nanu Safakatullah, ma'am. Yes, sir. Okay, sir. Please share your slide.
So is it visible?
No. Yes. Please make a full screen.
Slides are visible.
Yes. Very much visible. Very much visible. Yeah. Thank you. Please proceed.
Okay. Good afternoon. Myself, Dr. Nanu Safakatullah.
Please, Nanu Safakatullah, sir. Please wait. After that, next participant is Sarvender, sir. Kunwar Sarvender, sir. Please be ready. After that, your presentation. Thank you.
Okay. So my presentation is emerging role of probiotic bacteria in protection against colorectal cancer. So in introduction, the probiotics is a live microorganism which when administered or intaken, they're going to give a benefit to the individual. So this probiotic bacteria includes lactobacillus, lactococcus, bifidobacteria, pediococcus, streptococcus, and so on. So actually they got a special property compared to the normal microflora. So they can proliferate or colonize in the intestine. They don't have any pathogenic effect on the individual. They don't have mutagenic or carcinogenic reaction, and they are genetically stable. So that means they are not able to pass the resistant plasmid which is present in them to other organism. So this when you take the colorectal cancer, it is the third most cancer in the incidence as well as the mortality rate, and it's accounting for around 9% of overall death related to the cancer. So chemotherapy is the effective treatment for this colorectal cancer, but it got side effects like there is a bone marrow depression, skin irritation, alopecia, nausea. So several side effects will be there. So the prognosis for advanced colorectal cancer is poor, and hence the prevention is required to control the incidence of studies have confirmed that endogenous microflora they have effect on the onset of colorectal cancer. So this makes it reasonable to think that changing this intestinal microflora could influence the tumor development. So in this study, a large number of organisms have been identified, which have been isolated, identified, and submitted to GenBank. So for this presentation, I am giving a information about a single organism. So that is a Lactobacillus fermentum strain CM3. It has been isolated from cow's milk. It has been identified as a probiotic bacteria based on the different properties given by ICMR, that is acid tolerance, bile tolerance, bile salt, hydrolase activity, activity and other special property which shows that the present organism is a probiotic organism. So this organism has been used in the treatment of NN dimethyl hydrogen induced colorectal cancer in mice. So in the experimental design, so we have used the Swiss albino mice aged around 6 weeks, and they have been divided. So the experimental design is like this. So they have been divided into 5 groups. Group 1 is the just control diet without any supplement, and group 2 is the control diet plus the dimethyl hydrogen injection has been given, which is acting as a potent inducer for colorectal cancer. And group 3, treatment with probiotic organism as well as the injection. Then group 4, treatment with injection as well as the probiotic infusion. And the group 5, pre and post treatment. So this has been divided like this. Group 1 is the negative control, group 2 is the positive control, group 3 pre-treatment, that is lactobacillus fermentum has been given to the mice and then treated with the dimethyl hydrogen, that is for formation of cancer. And group 4 is the
Dr. Safakatullah sir, your voice is break.
injection colony form milk the so the following parameters have been studied. So these are the 10 different parameters which have been studied in the mice. So the first was general health status of the experimental mice. Second is food intake and body growth and survival study. So in the food intake as well as body growth and survival study, we have seen the increase in the body weight of the organism has been seen. Then there is the tumor formation. So in the tumor formation, we have seen that the negative control has not shown any formation of tumors. In the positive control, we got around 10 tumors, whereas the pre and post treatment, we got around less 1 or less than 2 tumor formation. So this is the picture of the presence of tumors. Then we have done the histology of the tissues to find out whether there is a presence of aberrant cryptic foci, adenoma, or adenocarcinoma. So these are the pictures of this one, the histological pictures. Then the fecal microbial count has been done to find out whether the lactobacillus, whatever we have given to the mice, they're still alive in the organism or not. So these are the uh, results where the increase in the concentration of lactobacillus has been found in the fecal matter compared to the coliformic bacteria. So the coliforms are those which are responsible to cause the cancers in the mice. And the fecal pH has been determined. So it is seen that the feces which where the acidic pH is responsible to decrease the formation of cancer, whereas alkaline pH of the fecus material was responsible for the formation of cancer. Then antioxidant activity has been done. So antioxidants are the substances that can prevent or slow damage to the cell caused by free radicals unable to unstable molecules that the body produces as a reaction to the environmental stress. So in that we have studied about the GST, that is glutathione transferase. So we have seen there is increased activities of GST has been found in the mice. Then we have studied about the superoxide dismutase. It is also the antioxidant. So here also, compared to the positive control, we have seen increased activity of SOD. Then we have seen about the reduced glutathione. Here also, compared to the positive control, we have seen pre and post mice have shown increased activity of this reduced glutathione. Then we have studied about the bacterial enzyme activity. So the fecal enzyme, that is the beta glucuronidase as well as the beta glucosidase have been implicated in converting the procarcinogens into carcinogens. So we want to find out whether this carcinogen converting enzymes are present in the mice or not. So we have studied about the glucosidase activity. So that we have seen that in the treatment group, that is the pre-post and pre and post group, there is decrease in the beta glucosidase activities has been seen. Then the beta glucuronidase activities has been studied, and in the treatment group we have seen reduced activities of beta glucuronidase enzyme. So in the conclusion, you can see the probiotic confer anti-carcinogenic effect by excluding the pathogenic microorganisms, by competing for nutrients and receptor, producing antimicrobial metabolites, in turn activating the host immune system. So these results suggest that the lactobacillus fermentum strain CM3 so could be used as a potential probiotic strain to control the colorectal cancer chances. The isolate has shown promising and significant potency in eradicating the dimethyl hydrazine induced tumor in experimental mice models. So, however, the successful translation of this preclinical strategies into clinical practice will depend upon the outcomes of clinical trials. The strain should be subjected to whether requires prove its stability for clinical and therapeutic applications. So in recommendation, there has been very less research done on the probiotics from non-fermented food sources. So it's also necessary to explore the probiotics from unusual sources. These potential probiotics should be characterized and commercialized. So very less probiotic supplements are available in the market. So since the potential probiotics got their main role in maintaining the intestinal health, reducing cholesterol level, improving the immunity and anti-carcinogenic properties, they should be intaken regularly for general health. So these are the few references, and thank you. Yeah.
Anyone want to any question? Sir, I have one inquiry.
Yeah.
What is the percentage of C3 is better than DMH? I hope DMH is commercial and
DMH is
CM3 are
commercial
used
one.
you.
Yeah.
Yeah.
So
So how
DMH
much
is
better?
commercial. That yeah, since DMH is having more potency to cause this colorectal cancer, we have induced with DMH only.
Okay. So which one is better? More?
So according to the literature survey, I can see that DMH is better.
But you use CM3.
So that is a probiotic organism.
Okay. Actually, your sound is very much break. Okay, thank you. Next participant is Kunwar Sarvender.
Yes, sir.
Kumaon University.
Yes, sir. Yes, sir.
Sir, are you ready?
Yes, sir. I'm ready.
Yes. Please share the PPT. Karisma ma'am, please share the PPT of Kunwar Sarvender, sir.
Yeah, thank you.
Hello. Is it visible
Yes.
now?
Can you rotate your mobile? Karisma ma'am, can you rotate your mobile?
Yeah, sure. Just wait. Huh?
Maybe slide is. Yeah.
Now
Very
is it
much.
okay?
Yeah. Very much. Very
Is it
much.
okay now?
Yes.
Okay,
Is
all
it
right.
okay?
Hello. Can I start, ma'am?
Yes, please.
Thank you, sir. Hello, everyone. I am Kunwar Sarvender, and my topic is the genomic profiling of the Sophocarpus tetragoniolobus. This is a type of underutilized legumes and belongs to family Leguminaceae. As we know that there is a very crisis to the protein-rich protein-rich compound to eat the people. and climate change and population is also increasing day by day. So there is a requirement to include in our diet the other sources of the protein. So this is a climbing in nature and is a protein content like soybean. The percentage of protein is 31 to 40%, and fat content is approximate 20% like soybean. And this ma'am, can I explain in Hindi, ma'am?
Yes, yes, please, please.
थैंक यू सर. ये जो है सर, इसमें ऑयल कंटेंट और जो है प्रोटीन कंटेंट काफ़ी मतलब ज़्यादा मात्रा में पाया जाता है. आजकल जो है प्रोटीन के खाने के सोर्स भी कम है, जो कि जो वेजिटेरियन है उनके लिए तो काफ़ी प्रॉब्लम होती है प्रोटीन रिच खाने में. और ये जो है मीट का अल्टरनेटिव भी हो सकता है. और इसमें ऑयल कंटेंट जो है 20% सोयाबीन के करीब होता है. और इसमें उसके केवल इसका जो ड्रॉबैक है, इसमें कुछ एंटी न्यूट्रिशनल फैक्टर्स होते हैं, बट जिसकी वजह से लोग इसको थोड़ा अवॉइड करते हैं खाने में. तो हमने मतलब जो प्रोसेस हमने यहाँ किया है, उस एंटी न्यूट्रिशनल फैक्टर्स को कुछ प्रोसेसिंग टेक्निक से कम भी किया जा सकता है और इसको जो है हम लोग कंज्यूम कर सकते हैं. तो इसका जो ऑयल कंटेंट है, हमने उसको निकाल के उसको इन विवो स्टडी एनिमल मॉडल पे भी किया गया और उसकी टॉक्सिसिटी भी चेक की. तो हमने देखा उसमें कोई मतलब टॉक्सिसिटी नहीं है. इसको इजीली मतलब लोग कंज्यूम कर सकते हैं. और जो यहाँ पे जीनोमिक प्रोफाइलिंग का जो मेन हमारा स्टडी का फोकस है, जीनोमिक प्रोफाइलिंग हम इसलिए कर रहे हैं कि ये मार्कर बेस्ड स्टडी होती है. और हम मतलब ये जैसे FLP, RAPD, ये डिफरेंट SSR मार्कर, डिफरेंट टाइप के मार्कर होते हैं जिससे हम उसको उसकी डाइवर्सिटी चेक करते हैं उसकी जीन की. तो ये अपनी स्टडी में हमने ये जो एक नई टेक्निक आई है नेक्स्ट जनरेशन सीक्वेंसिंग की, तो उससे हम जो है SNP के बेस पे जो है हम अपनी जो है प्रोफाइलिंग, जीनोमिक प्रोफाइलिंग करते हैं. और हम उसमें ये देखते हैं कि वो कौन सा जीन है जो जिसकी वजह से हम उसमें कुछ म्यूटेशन या कुछ वो करके उसकी क्वालिटी को और इम्प्रूव कर सकते हैं. तो इस स्टडी में हम जीनोमिक प्रोफाइलिंग में इसको टोटल SNP हमने इसमें मतलब डिस्कवर किया है, जो अभी डाटा इसमें नहीं दिखाया गया क्योंकि वो अभी अनपब्लिश्ड है, तो इसलिए उसमें नहीं दिया गया है. तो वो जो टोटल जो है SNP हमने इसमें डिस्कवर किया और उसको सारे SNP को हमने उसकी ऑयल के साथ जो एसोसिएट करके हम ये देखें कि वो जो ऑयल यील्डिंग कैपेसिटी वो कौन सी जीन रिस्पॉन्सिबल है जिससे मतलब फार्मर या लोगों को जो है अच्छी यील्ड जो है मतलब मिल सके. और ये जो ये फैटी एसिड, इसमें ओलिक एसिड का कंटेंट काफ़ी करीब 36 से 40% इसमें ओलिक एसिड होता है, जो कि मतलब सोयाबीन के इक्वल होता है. और इसके ऑयल की कुछ एंटी माइक्रोबियल एंटीऑक्सीडेंट एक्टिविटी भी होती है. तो ओवरऑल ये मतलब कोई इसका टॉक्सिक इफेक्ट नहीं है. और इसको जो है हम जो है मतलब एक्सप्लोर करने का मतलब कोशिश ये किया जा रहा है, क्योंकि ये अभी ये बीन टाइप का है और बहुत कम ये लोगों को इसके बारे में इंफॉर्मेशन है. तो इसकी मतलब उद्देश्य है ये इसके लिए किया जा रहा है. बाकी ठीक से, थैंक यू.
Thank you so much.
Okay, sir. Thank you.
Any question? Next invite delegate is Dr. Bavita Malik, Department of Zoology, Mukund Lal National College, Jamuna Haryana. Dr. Babita Malik, ma'am.
I'm sharing my. Okay, you share it. Hello, sir. Are you hearing me?
Yes, very much.
Good afternoon, respected chairperson, jury members, my dear teachers, scholars. I, Babita Malik, Assistant Professor of Geology, Mukan Lal National College, Yamunanagar, Haryana. I'm here to present my research topic entitled A Study on Fishbone Parasite in Fish Culture System in India. India is the second largest fish producer in the country in the world, and it holds second largest aquaculture producing country in the world. So fishery is very important to raise the economy of India as it provides employment, protein rich food and foreign exchange earnings. There is total fish production is about 10.07 million metric ton, out of which 65% is from inland sector and nearly same is from the culture system. So during the 2015-16, there is about 1 lakh crore value of fish production for local consumption export in India, and fish farming is very important. So there is also fish farming is at high risk investment due to fish diseases, and these diseases are generally caused by the fishbone parasite. Fishbone parasites are helminth parasites which broadly divide into two main categories. One is the flatworm and second one is the roundworms. The flatworm are further divided into two categories, trematodes and cystodes. Then cystodes are called tapeworms and trematodes are generally called flukeworms. Flukeworms are further divided into two categories, that is monogenetic trematode and digenetic trematode on the basis of their host during their life cycle. Next slide please. Diagenic trematode are the most diverse group out of these two categories because they have their complex life cycle. Roundworms are subdivided into two categories, that is nematoda and acanthocephala. Acanthocephalans are thorny-headed worms, and there have been several investigations have been done in the India also worldwide, and fishes are the host for many helminth parasites and for their larval forms, the adult of which are generally found in amphibians, reptiles, birds, mammal, and predatory fish. So there is a strict dependence on the parasite of its host, and their exploitation of the host by the parasite provide a useful research model in the field of ecology and evolutionary biology. Next slide, please. So these parasites directly cause economic loss as they cause reduction in the fish production. So several helminth parasites remain a subject matter for major public health concern, mainly in Asia, as these can be transmitted for fish to the higher vertebrates, including humans and domestic animals. So therefore, the objective of present study is to aware and educate people about the zoonotic risk of the fish by eating undercooked fish meal. Next slide, please. The present study was carried out at Geology Department, Mukan Lal National College, Yamunanagar during the time period April 2022 to September 2022. Next slide, please. So the present discussion, the present investigation deal with the fish-borne parasites present in the fish culture or the aquaculture system. So main cause of this fish-borne parasite disease are scarcity of potable water, consumption of raw or undercooked food, lack of personal hygiene, poverty, and favorable climatic condition for parasite growth and transmission. And other reason for these parasitic infection is direct dermal exposure of infective stage of these parasite to open wound or abrasions during handling of infected parasite containing fishes or even infected water. Among these fish fish-borne parasites that affect the freshwater fish. The helminths are mainly divided into 5 taxonomic categories. I already told that are trematode, monogenic or digenic, cystoda, nematoda, acanthosyphola. Worldwide, they are causing veterinary problems, and Trematoda, Cystoda, and Nematoda. These are the 3 helminth phylum which are responsible for the fish-borne parasites mainly. The phylum Nematoda include fish-borne parasites of family Aniscadidae and Gnathostomidae. Similarly, the phylum Cystoda parasites include member of the single family Diphyllobothrium Bothridae, an example of which is Diphyllobothrium, and digenic trematode parasites include member of the family Ophisthorchidae. An example is the Ophisthorchis, and Heterophilidae family member is Heterophilides. Heterophiles. So, the disease due to these parasites is the single most important factor which is threatening to the fish industry worldwide in an aquaculture system. Next slides, please. At least 50 species of helminth parasites have been implicated as producing zoonotic infection resulting from eating raw or undercooked aquatic or freshwater fish all around the world or even in India. These parasites cause disease in fish of the fish culture system, and nematodiasis, anicasis, gnathostomiasis. These are the common diseases generally found in culture system fishes. Monogenian trematodes have a specific organ for attachment to the host fish, and that is called hapters. And the most famous example of these are Gyrodactylus and Dactylogyrus. These are 2 trematodian parasites which affect the skin and gill of the parasites. Gyrodactylus affect the gills, therefore it is called the gill fluke, while the Dactylogyrus affect the skin of the host fish, so they are called skin flukes. They generally make a whitish color cyst on the skin or the gill of the host body and cause fading of the color of the host fish. Next slide, please. So, on the basis of color of small pimples on the skin of epithelium of the host, the digenic trematode cause disease, termed as white, black, or yellow grub. Grubs is the disease which may be white, black, or yellow. It is found generally in catfish, including Claris batrachus and Heteroneustis foliaceus during summer period in a culture system. But no mortality rate is recording to these diseases, and these are easy to control just by cutting the tree around the around the aquaculture system, and by making the culture system free from the fish eating bird, it is possible to control these parasites. This method is generally used in northern India. The ancestral parasites are called tapeworms. These are generally found in their larval forms only because birds and mammals are their final host of these cystodes. And example is Lingula intestinalis. As the name suggests, it is found in the intestine of the host fish. And host fish is Catla catla. So it causes lingulosis disease, which symptoms are sluggishness and slight distension of the abdomen. Then next phylum is the Acanthosypholan. The fish-borne members of the Acanthosyphola are thorny-headed worms. These parasites generally inhabit the alimentary canal of their host, and there is no specific symptoms is exhibited by these, or these are generally of educational interest. So these parasites use their hook to attach with the host intestinal epithelium, and example is Acanthogyrus and Palicentenis. These are two acanthocephalans which are mainly harm the major Indian carps. Some example of the fish-borne parasites are as under. First is the Eubothrium. Eubothrium is a cystode parasite, and it generally block the intestine of the intestine of the host fish and result into the gut blocking. So there is mechanical damage to the gut. Symptom is the mechanical damage to the gut. Then next parasite is anisakis, and this cause disease anisacosis, and this is a nematode parasite. It is an important fish-borne parasite because it causes gastrointestinal and allergic symptoms in the host fish. And next slide, please. Thank you. Next slide, please. Next fish-borne parasite is Diplostomum, and this parasite is a trematode parasite. It causes disease diplostomosis, and symptoms are black spot found on the body, and these are easy to cure generally by using the salt water bath. Next slide, please. Gyrodactylus and dactylogyres, I already told about these, and their treatment is very effective by using 5% salt solution given for 5 minutes in the initial infestation of these parasite. And second method is alternate bath in 1:2000 acetic acid solution followed by bath in 2% sodium chloride salt solution is very effective method. So I conclude that these fish-borne parasites affect the survival and growth rate of the fish in an aquaculture system, which directly responsible for great economic loss in aquaculture system, as these chemicals used to control these parasites are very expensive in market. Therefore, to alleviate such losses, it is crucial to take precautions to prevent such fish-borne diseases and such parasite-borne disease and reduce pathogenic level in culture system. As these parasites are zoonotic parasites, so the fish farmers are not aware about these parasites. So proper awareness programs should be maintained to educate the farmers about these parasites. Next slide, please. Hence, it is necessary to give basic knowledge about the common fish-borne parasite and disease caused by them and their control measure to fish farmers so that it will help them to treat the fish diseases and increase fish production in their culture system. Next slide, please. So, there is some preventions and control method. By using those, we can control the population of these fish-borne parasites, and it includes maintain a good culture environment and prevent deterioration of water environment. So next slide, please. Use hygienic and nutritious fish food to boost resistance of the fish stock and minimize the change of introducing pathogen of the water body. Third is maintain a suitable stocking density. Fourth is encourage fish farmers to join the fish health inspection program and conduct a simple health check on their fish stock every day. These are some references, and thank you so much for listening me patiently. Thank you.
Thank you so much, ma'am.
Thank you, sir.
Any koi question? Anyone want to ask any questions? Please raise your hand. Yes, sir. Yes, yes, Ajay Kumar Gautam, sir.
Ma'am, can you define monogenetic trematode and digenetic? What is the difference between digenetic and monogenetic trematode?
Sir, actually monogenetic trematode has single host in their life cycle, and digenetic trematode have two hosts in their life cycle. Therefore they are called digenetic. Secondly,
What is the host of digenetic trematode? What's the name of digenetic trematode host? They completed their life cycle on two alternate.
Hanji sir, please.
Digenetic trematode completed their life
Monogenetic
cycle
trematode
on
is
two alternate.
gyrodactylus. Hello, Hanji.
Digenetic trematode completed their life cycle and two alternate. What is the
Actually,
name
sir,
of two alternate?
they are viviparous. They give young one. They give birth to the young one, and example is gyrodactylus. And digenous trematodes, I already give the example in my slides. That is
Okay, good.
Diplostomum, sir. That is digenetic trematode. Thank you.
Thank you, sir, and thank you, ma'am. Our next participant is Dr. Ridhima Arya, Department
Yes,
of Agriculture, MMDU Mulana.
so should I share my screen?
Yeah, please.
Sir, are the slides visible?
Yes, very much visible.
Okay,
Please.
so I'll start. Here I'm going, good afternoon to all of you. I'm going to present on the precision agriculture, a topic very much in demand nowadays. Precision agriculture is an integrated information and production-based farming system which is designed to increase the long-term site-specific complete farm efficiency, productivity and profitability while reducing the impacts on the wildlife and environment. Now, as we all know that agriculture is the backbone of our country and economy, which accounts for almost 30% of the GDP and employment rate at 70%. So it shows a rosy picture of our agriculture, but how long it will sustain the growing demands of the ever increasing population, and this is a difficult question to be answered if we depend only on the traditional farming. So to meet the upcoming demands and challenges, we have to divert towards the new technologies for revolutionizing our agricultural productivity. So precision agriculture requires special tools and resources to recognize the inherent special variability associated with the soil characteristics, the growth of the crop, and then to give the appropriate management strategy at a specific basis online at the site. So we have three components of the precision agriculture: the information or the database, technology, and the management. Information or the database include the collection of the studies about the soil, crop, and the climate. For soil, we access the soil texture, structure, physical condition, moisture, and the nutrients present in the soil. For crops, we access the plant population, the crop tissue nutrient status, the crop stress, the weed, if they are the weed patches, the insect or fungal infestations, and the yield of the crop. For climatic conditions, we access the temperature, humidity, rainfall, soil radiation, and wind velocity. So bringing us small details, soil texture indicates the relative content of the particles of various sizes such as sand, silt and clay which combine to form the soil. Texture influences the ease with which the soil can be worked out for the agriculture, for the growth of the crops, the amount of water and air it can hold, and the rate at which the water can percolate through the soil. The soil structure describes the arrangement of the solid parts of the soil and of the pore space located between them, and can be determined by individual soil granules which clump, bind together, aggregate, resulting in the arrangement of the soil pores present in between them. The physical properties of the soil which are measured include the color, texture, structure, density, porosity, etc. The colors of the soil, as we know, they vary widely, especially in India, such as the important, and it gives the indication of the important properties such as organic matter, water, and the redox conditions. The soil moisture determines the efficiency of the plants and also regulate the species diversity, whether they are the plants, the animals, or the microbial complexes present surrounding those plants. Soil is a major source of nutrients, as we know, which are required for the growth. The three major nutrients, NPK, which are generally added, they make up the trio called the NPK and also named as macronutrients. The other nutrients required are micronutrients such as calcium, magnesium, manganese, zinc, sulfur, etc. For the crop, we do the assessment of the plant population, which refers to the number of plants per unit area of the land. And plant spacing, it refers to the arrangement of plants on the area planted. Since we maintain a referred gap between the two plants so that they get the sufficient sunlight and sufficient moisture and nutrients from the soil. The plant tissue analysis is carried out in the lab, which defines the elemental content present in the complete plant or the parts of the plant, especially in the leaves or the petioles. In the field crops, if we do the plant tissue analysis with the soil test programs, we can check out how much of the fertilizer amount is required for a particular plant species at a particular area at a particular rate. Now, as we know, crops, they experience an assortment of environmental stresses because agriculture is an open system. Like there is effect of the both abiotic conditions like drought, waterlogging, salinity, extremes of temperature, high variability in radiations, as well as the effect of the biotic insects, birds, other pests, weeds, pathogens, etc. The ability to tolerate or adapt these changes by effectively countering these stress is a multifaceted phenomena of the plants. Now, weed patches, like the type of the weeds are identified, their intensity is included, and all those studies are carried out within the field so that we can identify the types of the weed and their properties. Now, similarly, their infection of the insects or the bacteria or the fungal infestation, they can be identified with the help of these new technologies. The crop yield is measured as the amount of the crop which is harvested at the end per area of the land. So the technologies which we use in the precision agriculture are the very newer technologies. These include the use of the hardware, software, and various new equipments, farming equipments. The first one is the global positioning system, which you all have heard of. GPS receivers are there. We can identify our position in real time. We can send our details in real time. Similarly, now we have prepared the implements which are connected to the GPS receiver. We can carry them directly to the field, or we mount them on the implements or even on the tractors and allow them to move around the different areas in the field, and those studies are then returned back at specific locations. Like these are the implements which are carried on to the soil. So we carry out the soil sampling with the help of the GPS. It's that simple method is there. The GPS is used to navigate the accurate sampling locations. Now, if we do this year after year, we have the repeatable soil research. We let to identify that how the changes are occurring during the growth per year in the soil. So we can identify these methods like that. There's a composite soil sampling. In composite soil sampling, we identify the like areas, like those areas where generally we add the moisture conditions, the climatic conditions are similar. We identify those areas. We call them the management zones. Now these management zones can be used to identify the variability in the soil for the micronutrients, the stresses which affect on the crops. All those studies can be carried out. There's another method which is a very useful method. It's called the grid sampling. Grid sampling is just like the mass sampling. So there are no like areas identified. Here we identify the different zones present in a small amount on every GPS point. We identify the number of samples and how those samples are taken that affect our fertilizer recommendations and the application of the fertilizer within that area, which will finally affect the crop yield. Now, another type of sampling which is usually preferred is the management zone sampling. In management zone sampling, a zone of like areas identified which responds similarly to the inputs like that of the fertilizers, seeds, the water, and the pesticides which are used. So if they reply in the similar way, they respond in the similar way, those zones are defined as the management zone. So we divide a field into the multiple similar areas, and these management zones are then worked out to identify the variations in the soil micronutrients, the stress on the crop, the growth of the crop. All these things are identified, and then efficient application of inputs can be done. Now, there are variable applications of GPS in agriculture, like we can identify the planting ratio, how the spacing should be done between the two different plant. As we now know, there is integrated plant management systems which are done that we grow two or more crops together. So we can identify the spacing between those crops and we can manage the growth of the different crops within the same field areas. We can create the yield maps for year to year changes in the yield which is coming from the soil. Then we can identify in advance that when the crop is going to be ripened, and we can plan our harvesting system, we can plan our harvesting areas. Similarly, we can identify the yield map, we can control the application of the herbicides and pesticides, as we can add the application of only those amount of pesticides which is required, and no more than that should be added, so that it cannot be continue to get magnified. Then we can plan the farm according to that. We can map the field and the yield, and also can use the farm machinery according to that. Now, for the this GPS can also be used to assess the availability of water in a particular area, that how much water resources are available, and we can use that to identify how much water can be gone, the canals, the smaller rivers can be made to divert towards those areas. Similarly, we can identify the waterlogged areas and can check out that which type of the plant can be grown on those waterlogged areas, rather using the simple crops. Now, there's also application of the geographic information system GIS in the agriculture. It includes the use of the computer hardware and software, which uses the different attributes and location data to produce the map. An important function of agriculture GIS is to store layers of information such as yield, soil survey maps, remotely sense area, crop scouting reports, and soil nutrient levels. A GIS system is a computer-based system which is used to capture, store, check, and display data related to positions on the Earth's surface. Now, GIS usually give us results in the form of different layouts. We can identify the areas where the wetland are there, the land covers are there, the landscapes are there, where's the location of streams, what kind of vegetation can be grown, and the different kinds of soil present over there. It includes the information about the different factories, farms, schools, etc. We can all identify through the basis of the GIS. It's a digital data which can be entered into the computer, and we can identify how we can modify the application of various inputs in the field. Now, GIS allows to give the multiple layer of information over a single area. We can identify which is the requirement of particular crop from that area. Similarly, technological innovations and geospatial technology, this help in creating the dynamic and competitive agriculture which is protective of the environment and capable of providing the excellent nutrition to the people. There are various applications of GIS, like we can identify the crop map, we can give the crop assessment about the crop health, the irrigated landscape, soil and irrigation systems, the agriculture mapping for the vegetation cover, the damage and land degradation is occurring over there, the assessment studies and the elevation models for the drainage. The next important technology which is now in demand is of the remote sensing. Remote sensing is a tool which is integrated into the GIS system. It uses the imaginary data. As we know, like the sunlight is coming to the earth, so it's absorbed by all the matter present over earth and then is reflected. So this light is measured, the reflected and emitted radiations are measured through a distance from the satellite or from the aircraft, and these images then camera which collect are studied out and help researchers to sense the things on the earth. The cameras on the satellite and airplanes, one second. Ma'am, मेरी presentation चल रही है, please come. धीरे. Cameras on the satellites and airplanes take images of the large areas on the surface, which allow us to see much more than we see on the ground. The sonar systems on the ships are also used. Sonar systems can help us create the images of the ocean floor without even touching the bottom of the ocean, since sonar systems of the variations of the different remotely sense data provide a tool for evaluating the crop health. The plant stresses can be identified related to the moisture, nutrients, compaction, crop studies, and other plant health concerns. Electronic cameras can also record the infrared images which are correlated with the plant health tissue. Since the photosynthesis, it's occur in the infrared region, so we can identify the how healthy the plant tissue, how healthy the photosynthesis is going inside the plant. So remote sensing, it can reveal also the in-season variability which affect the crop yield, and so we can make the timely management decision to improve our profitability from the crop. These images can be used to develop and implement a spot treatment plant system which optimizes the use of different chemicals like pesticides and fertilizers. We can accurately add the amount which is required, and not more than that amount should be added in the crop. So there are different satellite images which come from the Landsat or spot. They can be used to generate the different maps of the particular fields, and we can identify the requirements of that field. Thank you.
Thank you so much, ma'am. Now actually our next session chair, Dr. Mallika Pathak ma'am, already joined our session. But sorry ma'am, actually only one participant are left and our session is late by 10 minutes. Ma'am, if permit me, then this is the last presentation.
Siddharth, no problem. Please continue.
Yes, ma'am. This is the last presentation, ma'am. After that, session will start.
Sure. Thank you.
Thank you. Our next invite delegate is Dr. Bipin Bihari Misra, sir, from Patna University, Patna.
Hello, sir.
Yes, sir.
Hello.
Yes.
I'm audible, sir?
Yes, yes. Very much audible.
Okay. Okay, sir. I'm sharing my screen now, sir.
Yes.
Hello, sir. My screen is visible now, sir?
No, sir. Not visible.
Hello, sir. My screen is visible now, sir?
No, sir. Your screen is not visible.
Now it's visible, sir. Hello.
No, sir. Your screen is not visible. Sir, may you try?
Yes, I'm trying, sir.
Sir, if create any issue, then I will shift your time at 2:30.
Okay, sir. No problem.
Okay. Thank you so much, sir. Now our next lecture will start. Guest speakers, and session chair is Dr. Mallika Pathak ma'am. Dr. Mallika Pathak ma'am, professor and head Department of Chemistry, Mirinda House, Delhi University, New Delhi. Dr. Pathak ma'am is an alumna of Mirinda House. She has been a faculty at Mirinda House since 2006. She is a recipient of award like Most Promising Innovation in 2016 from University of Delhi, Teaching Excellent Award and Innovation in 2015 from University of Delhi, and Australian Leadership Award Fellow ALAF in 2013. A computational chemistry by training, she has many research papers in various peer-reviewed international journals, authored one book chapter from Elsevier Publishing House, and filed a patent in 2017. She is passionate about promoting active learning and working with students on innovative projects. Her research interests including drug molecular interaction, nanochemistry, and its application to water purification, etc. She has been a PL and Copper PL for 3 major research projects under Delhi University Innovation and Star Innovation Project. At Project C is the teacher in charge, Department of Chemistry with effect from February 2019. Ms. Mallika does research in chemoinformatics, biochemistry, and chemical biology. The current project in pharmacological properties assessment of ethyl pyruvate. She is also a life member of Indian Biophysical Society, Green Chemistry Network Center, Chemical Research Society of India, and Indian Society of Analytical Scientists. So now I warmly welcome Dr. Mallika Pathak ma'am. Please, ma'am.
Namaskar, everyone. Thank you so much, Siddharth, for this kind introduction. It's a little big, but doesn't matter. Thank you so much. And I welcome everyone to this conference and congratulate the organizers for organizing such a lovely international conference. Many congratulations to you and all the best. Siddharth, would you like to call the first speaker now?
Yes, ma'am.
Please go ahead. Who is the first speaker?
Ma'am, Dr. Sushil Kumar Upadhyay, sir.
Okay, so most welcome. Dr. Sushil Kumar Upadhyay, most welcome. Siddharth, would you introduce sir to us?
Yeah, sure, ma'am. Actually, Dr. Sushil Kumar Upadhyay, sir, is working as an associate professor in the Department of Biotechnology, MMDU Mulana. Dr. Upadhyay has 12 years of teaching cum research experience in the field of parasitology, taxonomy, molecular biology, animal and veterinary science, environmental biotechnology, bioecology, nanotechnology, entomology, and microbiology. Dr. Upadhyay is recipient of the degree of Doctor of Philosophy in Science for University of Allahabad. He received undergraduate and postgraduate degree from Dr. Ram Manohar Laria Award University, Ayodhya, Uttar Pradesh. Dr. Upadhyay has received more than 2 dozens of awards, including Certificate of Excellence, Bharat Jyoti Award, Bharat Shiksha Gaurav Puruskar, Certificate of Excellence. Professor A. Sushil Yugomethi Parasites Taxonomy Promotion Award, Professor S.P. Gupta Gold Medal, Professor S.L. Misra Gold Medal, Best Teacher Award, Pranvaran Sri Samman, Pranvaran Ratna Puruskar, Vigyan Ratna Puruskar, and facilitated many more fellow awards. His research skill handling of experimental phenomena is excellence and published 33 books and 155 national and international research papers in reputed, referred and high impact journals. He has attended and actively participated in 71 trainings and FDP and presented his research finding in 185 seminars conference at international and national level with outstanding scholar profiles, H index I7, I10 and 38. Dr. Upadhyay has no stone unturned in the field of molecular taxonomy and nanobiotechnology for helminthes and taxon validation at helminthes, which can be evaluated by his great contribution on DNA database and submission of 2 patents. His contribution for employment to youth at grassroots level for livelihood and sustainability by the mission transfer of science and technology to grassroots level and vision earn as you learn is also remarkable and cannot be ignored. Dr. Upadhyay has communicated his vision at individual level through mass communication, magazines, daily visions, daily newspaper and direct contact. Dr. Upadhyay is an ideal teacher and his hard work inspired the students to achieve their goal. Now I would like to invite Dr. Sushil Kumar Upadhyay sir.
Thank you, Mr. Siddharth.
Thank you, sir. Karisma ma'am, please share the PPT.
Okay.
Karisma ma'am, please share the PPT. Karisma ma'am, please share the PPT. Sir, please wait. Actually, network issue.
Okay, Siddharth, no problem. So let me start my talk, okay? And whenever it is possible, you have to start the slide and join at that time. Okay, so dear friends, as we are gathered here to discuss some recent approaches of recent trends in biotechnology and life sciences. Keeping in view of the principal theme, I just prepared a presentation of my work that is entitled to Biotechnological Approaches in Sustainability and Control of Parasitic Diseases. So there is two major objectives. One is the role of biotechnology in the sustainability, and the second, how we can use the biotechnological tools for the control of diseases.
Sir,
so
I
there
hope
are,
this is visible.
Yeah, very much visible. So I suppose there are several diseases which are called life-threatening diseases may not control directly by the application of drugs until unless we don't have a real causative agent, known about the real causative agent or aware about the real causative agents. So dealing to the real causative agents, either it may be microbes, it may be parasites, it may be other foreign particles, or sometimes it may be physicochemical factors like radiological factors, mechanical factors, and radiations. So come to the first objective, that is biotechnological approaches in sustainability. Our entire objectives, as I said, it is principally having two things, and the second is the validation using microbial and molecular tools and techniques. So next slide, please. As we are very much aware, and right now we are working on the bioentrepreneurship. Okay, so bioentrepreneurship dealing to the apiculture, sericulture, aquaculture, life culture, applying of the living organism to earn the money to raise our economy that helps in the sustainability, lifestyle, etc. So here our principal objective following through the apiculture. We principally and care our honeybees for principally one purpose, that is to obtain honey from the honeybees. And secondly, we try to obtain propolis that have certain medicinal values and economic significance. But the most important aspects that we don't care and try to neglect all the times, that is bee venom. So if we work upon the bee venom, then by the application of a little amount of bee venom, we can cure several life-threatening diseases, including metabolic diseases, physiological disorders, some epigenetic diseases, and the pathogenic diseases as well, especially the diseases caused by parasitic helminths. So we have designed a certain instrument in our laboratory, which is few of them has been patented by the School of Parasitology, University of Allahabad, and some under the process of patent, even though has been published under the observation right now, to extract the crude bee venom and thereafter try to process it using lyophilizer and mass spectroscopy. So this is the instrument, is the lyophilizer. That is the process of freeze drying method by reducing the temperature and creation of vacuums to absorb all kinds of moisture available in the bee venom that may help in the longer term applicability of the bee venom. And after processing through the lyophilization, we proceed for the next aspect, that is called mass spectroscopy. And mass spectroscopy helpful in the molecular diagnostic, or you can say that the constituent level of the bee venom. And after following the ideal tools and techniques of the mass spectroscopy, or by the processing of the sample, we obtained it in the form of certain graphic representation peaks, and that will further evaluated it in the form of certain aspects. So next slide, please. And by the mass spectroscopy, there are some key elements has been recognized, which the combating of several vector-borne diseases, parasite-borne diseases, and other life-threatening diseases. In this includes two major things, that is gelatin, and the second is phosphokinase. Both these things are helpful in reducing several diseases, including hypertension, blood-related diseases, epigenetic diseases, vector-borne diseases. And these kind of aspects has been communicated to our society, make them applicable and make them aware about the application of bee venom that is neglected by our community, our farmers, and somehow our scientific communities as well. Through several workshops, training, symposia under the umbrella of non-government organizations, along with under the sponsorship of government organizations like UGC, DBT, INSA, and university itself giving certain promotions to work upon these aspects. So next slide, please. Mr. Siddharth, carry on to the slide number 7. Yes, this is the awareness program we continuously working upon to make the aware about our society persons, farmers, and at grassroots level. And during this work, whenever we're trying to make them aware, we also incite something about the self-sustainability by creating self-help group, so that the unemployed youths, women living in our house, in our society, who are not directly working upon any organization to gain the economy, they can, by creating self-help groups, by utilizing the natural resources without any payment, and they prepare the honeybee boxes so that they will be able to cultivate the honey, and then by providing the epitoxin extractor by us, they are able to extract the bee venom. And this bee venom is very costly material that is sell at the level of 10,000 per gram by the and purchased by the pharmaceutical industries, which are under the MOUs with us, that working with us in a networking collaboration things. So the first objective has been attained by going through this, and the second objective is the parasites and parasitological diseases. So next slide, please. Since last 10 years, I'm working on freshwater fisheries, marine water fisheries, especially in terms of parasitic investigation to find out the hidden fauna, microfauna communities living inside the marine fishes, freshwater fishes, and affecting their lifestyle, physiology, bioecology, and ultimately the productivity in terms of the quality and quantity. So the exploration of these biotic fauna will be helpful in the management of aquaculture. And by going through the freshwater fishes, several catfishes, freshwater garfish, in the marine fishes, sharks, silver whiting fish, Silagosihama, and other deep sea fishes, we have reported several worms coming under the platyhelminths category, like cestors and tremators. Both are the endoparasitic, exclusively endoparasitic worms, and nematodes, that's called roundworms. And these were brought to laboratory, process it for photomicrography, and process it for molecular diagnosis, and process it for numerical taxonomy. The numerical taxonomy were based upon qualitative divisive classificatory system proposed by a school of parasitology from Allahabad University, and molecular taxonomy based upon the working or based upon the analysis of conserved gene sequences that are very much persistent in the living organisms, and the alteration or changes in their sequences may lead to the origin of new phylogenetic clades, and it will be helpful. Even though if the persons are in schools having very much facility to work upon the molecular work, then they can go through the genome analysis. But if there is a lack of infrastructures, there is a little or constraints of the financial supports, then they can follow the conserved gene sequence analysis, and by going through these conserved sequences, that can analyze the correct taxonomic status of any living organism. The results obtained from the molecular analysis were further evaluated and validated by in silico analysis using some advanced, sophisticated softwares. Most of these are freely available, including Mega, Cluster W, Phylogeneia, that helps in the blasting, editing, and creation of certain dendrograms, cladograms, and phylogenetic analysis. So these are the materials and methods that were being followed for our research purpose. And by going through this, preliminary aspects, any students working in the PG level can show their interest in the parasitology at a limited resources. So next slide, please. While working on the parasites, we have noticed that there are several parasites whose secretions, metabolic products may causing certain alterations among the respective hosts. But there are certain organisms, parasites, which having capacity to transfer through the trophic level, from one trophic level to the another trophic level, either through the edible sources like foods, or sometimes through the direct contact or by drinking materials, and it may reach to the human being. So those parasitic organisms which have capacity to transfer from their lower trophic level to the higher trophic level up to human beings through the edible sources, drinking sources, or our natural sources called zoonotic parasites, and the transmission of these communities are called zoonoses. So all parasites are not so lethal for our especially, but those having zoonotic capacity, they are very much lethal and dangerous for us. So it is imperative to be aware about certain zoonotic parasites occurring in our neighboring animals, cattle, source of foods, aquacultures, and these are specially belonging to Contrasicum, Anisakids, Spiroidians, Trichuris, Trichinaris. And these aspects has been validated and evaluated by the experimental observations as well. So next slide, please. Next two slides. Move ahead by two slides. One more. Yeah, this is a freshwater garfish, Genetodon cancella, and it is reported from the river Yamuna. And it is very much efficient, delicious fish, and it carrying the Spirarian nematode that is designed as a Sturdinema multiembryonata, which is a newly designed and designated species of the nematode by our parasitology group, has been published in Journal of Species, International Journal with High Impact Factor in 2017. The multi-embryonated eggs of these worms fed to the control mice, and then after a period of observation, we have first of all noticed certain physiological alterations in the mice, and thereafter going through its sacrification, after following the rules and regulations of the ethical committee and guidelines, we find out certain developing phases in these mice model. So next slide, please. So you can find out the mice model carrying certain multi-embryonated eggs and their site of location or microhabitat or niche. And next slide, please. So as far as the morphotaxonomy is concerned, we can follow the conventional taxonomical method through the morphological observation using the compound light microscopic image, but there are certain image characters that were very tiny and may not be able by correct observation by using the compound light microscopy. So we wish to follow, or we recommend to follow, the ultra topographic imaging that will help in finding of more accurate shape, size, and number of the characters which are especially significant. This is the study mouth opening guarded by a collar. Body is entirely covered by sturdy spikes. These spikes are various shape and size. Some may be single pointed, some may be multiple pointed with common base. So based upon these occurrence and their characterizations, distribution of ampli, amphi, plasmids, and microns, we have ultimately able to diagnose the genetic level or generic level of these organisms. These findings were further substantiated by chimeral Isuda diagram. Next slide, please. That were handmade diagram using the aid of microscope. Sequence 14 chimeral Isuda made of Japan were used to create this diagram. So next slide, please. Line diagram. Yes, this is the line diagram or chimeral Isuda diagram of study Nema multi-embryonata based upon the microscopic observation using light microscopy. So next slide, please. The findings of the conventional microscopy showing that these are quite different from the pre-existing ones, but there is a need to further validate following through the molecular taxonomy, and in the molecular taxonomy, following the cladogram analysis, we have found that the never joining tree using 18S RNA and the conjugate gene sequence analysis COI has reflected that the newly recovered worms alighting are different from that of their earlier existing species like Arengescaris, Rhapidoscaris, Goizia, Terranova, Contrasecum, etc. So next slide, please. And these are making a different and new clades that is presented here by the bracket, red bracket, and this is showing all the organisms are falling under the single clade are aparted from the Goizia, but even though they are showing quite close correlation with the Goizia is comparison to the Ascaris, Toxoscara, Goizia, and the similarity between these Goizia and newly discovered depiction of the different findings. Some cytochrome oxidase, some are based upon the 18S RNA, and some are based upon the ITS gene analysis. But all kinds of findings showing a quite common similarity there. Spiroidians presented as here, study Nema multi-embryonata showing a different clade formation in comparison to the other pre-existing and valid genera of the nematodes. So these kind of aspects can be helpful in further diagnosis. There are several other nematodes, as I discussed earlier. They have also shown certain characters and presented here for a kind interest, but these are not the kind of the investigation or kind right now. And if we further follow, then we can find out the cestodes, and these cestodes, these are the microscopic analysis. Move forward. Next slide, please. Next two slide, please. Come to the cestodes. Yeah. So these are the cestodes presented here. Some are belonging to the tetraphyllirian cestodes, some are Sclerotinia, some are the tenuate cestodes. There are different characteristics you can notice by the number of hooks, orientation of hooks, number of the bothria present in it. All these things are the diagnostic characters of the genera. Next slide, please. These findings can be further investigated through the ultratopography. Next slide, please. And in which we have found the Rangesia, Senga, Lithocystus, and these are very much illuminative and presentable diagram, and it is helpful in the generic diagnosis of certain cestodes that were present in our surrounding vertebrates, and major of the things are reported from Mastacymbalus armatus, Senga, Polyoncobothrium, Rangesia, and Tinea has been reported from the wild rats. Sclerotinia has been reported from shrews in the our householding shrews. Lithocystus from the Clarius batrachus. And these are the other aspects that will be helpful in the validation at taxonomic status. So, Mr. Siddharth,
Yes, sir.
I wish to stop it here because we are already running quite late and the pupils are waiting. We can listen out to me at any time. We first try to listen our young scholars who have given the time at 2:30. So it is about to 2:30. So I would like to stop it here. Otherwise, it will take quite more time. Okay?
Yeah, yeah. Sure, sir. Sure, sir.
Yeah, we will work upon. So thank you so much, Siddharth.
Thank you, sir. Thank you. Thank you so much, ma'am. Dr. Mallika Pathak, ma'am.
Hello. Yeah, thank you so much. It was a lovely presentation and I learned quite a bit. So I would request one of you to pose a question to the speaker, if you can do that.
Yes, ma'am.
Yeah.
Sure.
Most welcome from the young scholars who are working in the parasitology or interested to work in the over the parasites are requested to please resolve your queries by asking some questions. I feel happy to share our views over there. Okay, Siddharth, over to you. Please carry on.
Okay, sir. Okay. Thank you. Now our next invite delegate is Dr. Bipin Bihari Misra, sir, Patna University, Patna. Bipin Bihari Misra, sir. Next presentation is Ravi Kumar in Mass DRDO. Next presentation, Mr. Ravi Kumar in Mass DRDO.
So my presentation is visible, sir. Hello.
Yes, yes.
Okay. Sir.
Hello, sir. Hello, sir. Radio. Hello, sir. Radio.
So a very good afternoon to all of you. My presentation is on the study on ameliorating effect of root extract of asparagus in alloxan-induced diabetes mellitus, one of the common concerned metabolic disease. So, since ancient times, plants have been an exemplary source of medicine, and Ayurveda and other Indian literature mention the use of plants in treatment of various human ailments and diseases. And India has about 45,000 plant species, and among them, several thousands have been
सर इसके बाद ही आपका ऐप ज्वाइन कर लेंगे। Dr. Bipin Bihari Mishra sir are left. Next participant, Dr. T. Lavanya. Dr. T. Lavanya. Next participant, Dr. Harpal Singh, Chandigarh University. Dr. Harpal Singh.
Listen me?
Yes,
Yes, sir. Good afternoon, sir.
Good afternoon. Miss Karisma, please share PPT. Miss Karisma, please share the PPT. Miss Karisma, please share the PPT.
I have already shared the PPT. Is it not visible?
No, it's not visible.
Is it visible?
No.
I try to share it. Is it visible, sir?
Yes, visible.
May I start now?
Hello?
Yeah, sure.
Yes, friends, good afternoon. Myself, Dr. Harpal Singh from Chandigarh University. I am working as Associate Professor here in Agroforestry under the Cluster of Sciences in Agriculture Department. So here I am going to give a presentation on socioeconomic profile, a tool for improving cultural and economic status of farmers from Sahibzada Ajit Singh Nagar, Mohali area of District Punjab. So first of all, a little bit about the talk that why we are going for this study. There are certain backgrounds. As we know that agricultural scientists improving crop plants, they are pertaining to different production, resistance to stress and easy to grow and harvesting of crops normally. So this agriculture is nourishing and proliferating the human population as such.
Dr. Harpal
Which
Singh,
will be
your slide is not visible. Just open the slide.
okay. I think there is some technical problem in this. Now it is visible, sir?
No.
No. I think there is some technical problem in MS Teams because people are waiting in the lobby. So kindly accept them, then it will be working.
No. Already.
Now it is visible, I think.
Yes, visible.
But it is control is not from my side.
No problem, sir. You just say कि next slide.
Okay. Just wait here. Okay, I was talking about the production, then adoption, then sustainability. So there are mainly three attributes for any sustainable management of crop. One is normally this production, this adoption, production, and sustainability. Kindly next slide, please. So as you know that poverty, hunger, unemployment, these are the burning issues. As we know that about 2 million people around the world suffer from hidden and hunger, and they are getting enough calories but not enough nutrition. So these are main focus points. So problem could be overcome either by providing supplements or by fortification of foods. So new technology will undoubtedly allow development of new types of nutritionally enhanced plants. So how these will be overcome? Next, please. Next slide, please. So what to conserve? Now there are three main components which are to be conserved. First of all, we have to conserve our soil, then water, then air. So if we go from the conservation of natural resources, so there are also 4 F's which are provided by our nature. For example, this forest is one of them. So forest provide us food, fodder, fiber, fertilizers. So these all are part of conservation. So for sustainable development, what we are going to conserve and what we can achieve in future, that are the main agendas. Next, please. Next, please. Next, please. So what are the different factors that are influencing for sustainable agriculture? Either we go for crop diversification, that means simply management of agriculture, management of horticulture produce, management of soil, management of different components which come under agroforestry systems. So several factors influence the implementation of crop diversification, which in turn contribute to the different dimensions of poverty. So the poverty condition influences across resources, which will influence the implementation of crop diversification. And ultimately, we have to interact with the stakeholders so that there could be solid these recommendations that would be applicable to the field simply. Next, please. Now, the ideal food system will only achieve from if there could be offering of adequate nutrition and health components, creating biodiversity and avoids negative ecological and environment impact simply, then ensuring livelihood for farmers, diverse landscapes, equitable access to land, water, seeds, and other inputs. So these all are important parameters which are trends in also agriculture system as well as in sciences. So technology, innovation, nowadays, as we can hear of this ideal system of those attributes of this IPR, intellectual property rights also, because in present era, patents, this trademarks, these have gained some more prominent importance. So these things are very important. Next, please. Next, this is regarding food technology. Next, please. I will come to survey part that is important. Next, please. Next, please. Now what are the different components which we have to achieve so that sustainable management will be that can be for further consideration. Now, if we go for simply growth and achievement of agriculture, are we creating our business? That is also an achievement. Or we are going for economy that will provide us power. Then there are different ecosystem services or products. For example, there are n number of minor forest products, n number of major forest produce that are providing our recreational activities. So number of tangible and intangible benefits are producing these natural resources which are giving birth to ecotourism simply. Then ultimately sustainable development can lead food security, which is one of the main component of this 17 sustainable development goals. So there are certain key attributes. For example, one is adaptability. That's going for diversification, culture, tradition, production, food security, innovation, trademark, entrepreneurship, food hub, IPR, that is leading to GI tags, and ultimately sustainability. Now, what can be sustainability? This consideration, is it social, economical, ecological? So these are main fundamentals. Next, please. Next, it is expressed horizon agroforestry. Next, please. Next, please. Next, please. These are different approaches. We all know that approach for diversification. This is well understood to all. I am coming only of this economic survey part. Next, please. Next. Next, please. Next. Now, kindly, no, back. Now, what are our present studies? Our present study was under RAVE program, in which our students, this study was conducted as preliminary information which is held from farmers. So under this program, in Kharad area, 3 different villages were selected on different questionnaire basis, and our study was conducted in Hasanpur village, Kallewal, Singhpura, and other adjoining areas. Farmers, cooperative societies, and other stakeholders were interviewed with a proper questionnaire, so that we had results were done interpreted accordingly. Next, please. These are the different villages from Google Lens. Next, please. Now, how to improve the livelihood status of farmers? First of all, we identify the problem of the farmer, then made a thorough interaction, then discussion, then better understanding and learning of the farmers from farmers. Then we conduct different farmers awareness and training programs, which was conducted with the university collaboration and KVKs. Next, please. Now, how to make solution? There were certain key backgrounds, whether we are going to create incubation center, agri startup, entrepreneur or agripreneurship, subsidies provision, GI tags, patent, trademark, IPR related issues, biocontrol, precision farming, organic farming, and either going for soil sampling. Though these are the parameters which can make a solution for the sustainable agriculture. Next, please. Now here are results. Some of the results are here. That among the farmers, now there are certain results which are very important. Present study was based on simply preliminary information which are carried under socioeconomic survey to assess the important information regarding agronomic practice, economic status, and livestock production of farmers. The study was conducted on the basis of proper questionnaire from different villages and adjoining areas. Nearly more than 200 farmers were interviewed for assessing their socioeconomic status, and in that status, students prepare different socioeconomic profile of the farmer on the basis of questionnaires. And in that survey, students collect the information regarding income status, caste system, education status, family composition, cropping system, availability of implements and machinery, availability of loans, source of information, etc. Each item was analyzed and explained clearly to draw meaningful inferences. Better health facilities, availability of better seeds, and provision of low-cost fertilizer are responsible for attractive farmers' status. And on interaction with all the respondent farmers, it has been found that literacy rate was very high, that is 90%, and further it was found that during Kharif season, majority of them cultivated rice, that is 80%, while in Rabi season, wheat was cultivated mostly 80%, and few of them cultivated sorghum and other minor millets for fodder production or use. And on an interaction, findings reveal that socio-economic status of the farmers can be improved by imparting technical knowledge to the farmers, increasing their education level, increasing their social participation, and further lack of awareness in the field of disease, pest management, farming inputs were also seen. Therefore, for the well-being of farming community, new technologies like disease monitoring with available software technology and artificial intelligence should be applied for sustainable agriculture. So in this study, we concluded that, next please. Next, please. Next, please. Next, please. Next, please. Next. Next, please. Kindly kept there. Now, what are the next future threats? Say, for example, we are going this low water table is one of the main important agenda. Scarcity of water, low water table, these are burning issues, and this strategy would be controlled with the help of new IITs which are coming in front. For example, at IIT Ropar, there is a project called Awadh, Agriculture Water Technology Hub, which is near about DST project, and it cost near about 3 crores. And those people are working for the betterment of the society. Next, please. Now, there are different solutions, but what are the main solutions? Main solution, for example, we are going for biosafety, awareness programs, attitude, behavior change, whether it is for farmer or it is for scientific community, learning by doing, adoption, production and sustainability, integration, infestation, market interventions, global demand, risk factors. For example, risk factors are anthropogenic factors, which cause major threat to the biodiversity. So these are burning issues which should be controlled, which should be minimized with proper policies, recommendations, and interactions of different communities. Traditional knowledge and other important agendas are also very important for that. So this is all about our study, what we have studied, conducted in different villages, what we have done. So for the solution, what we have done, we have done different campaigns. Campaign is related to tree plantation, minimize of different these chemical fertilizers or other fertilizers which are very harmful to soil also, plants also. So these are some of the beginnings what we are going to for globalization or for sustainable agriculture. So thank you one and all. This was all about our study. If there are any question, any recommendation, any suggestion, house is open for all the things. Thank you one and all.
Thank you, Dr. Harpal. May I ask a question, Dr. Harpal?
Yes, yes, ma'am.
All this data that you've generated and all, have you taken the data from the Government of India websites or your state government website, or have you done a survey for this?
Okay, ma'am, this is basically RAVE program, Rural Agriculture Awareness Program, which is done by the students itself. So, जो data की बात है, the data which is available on the website, that is secondary data. That is secondary data, and this data is primary data. So there is difference between primary data and secondary data.
No, that I understand. But so all of this work that you said that the students have done, so they've collected the data from the farmers
itself.
And the society itself. That's the
and
reason.
the society. Yes, yes, ma'am.
Right.
Yes, ma'am.
So how many respondents did you have?
Ma'am, there we have selected 3 different villages near Kharad. One was Hasanpur, one is this Kalyanwali, and other is Singhpura. Now you all know that this present situation is that maximum communities, maximum farmers, maximum people have shifted to other countries. इक्का दुक्का यहाँ पे रह गया है, तो ये बहुत टफ है, सर्वे करना बहुत टफ है. It is not an easy job because I was also with students. So that for the people to this make more interaction, what we have done campaigns, we have made several programs so that people will come and interact with the students. So it is not an easy job. It is a very this challenging job. So we are working on it. It is not, we have not closed it. It is just students have collected some of the data. So I get a report, then I present here. So it is sky is the limit. So this has not been closed. So your recommendations, your suggestions will be also incorporated for fruitful these studies in future also. Thank you. Thank you.
Many congratulations for this work and for thinking it and taking your students into the confidence.
Thank you.
If there is any questions from the audience, is there any question from the audience?
I hope no, ma'am.
Okay,
thank you so much, sir.
Thank you. Thank you.
Now I would like to invite you, Mr. Ravi Kumar, INMAS, DRDO, Defense Research and Development Organization, New Delhi.
Thank you, sir. I am audible.
Yes, audible, very much audible.
Sir, can I start?
Yeah. Please share.
My slide is visible to all.
No. No, your slide is not visible.
I think now this is visible.
Yes, very much visible. Yes. Please proceed.
Good afternoon, sir and ma'am. Myself, Ravi Kumar, Deputy SRF, working under the supervision of Dr. Rajkumar in INMAS. My research topic is N-acetyl L-tryptophan L-net attenuates radiation-induced oxidative stress and mitochondrial dysfunction in neuro 2 cells. As we know, radiation is present everywhere in this universe, and we continuously exposed to radiation which is comes from the natural backgrounds, and the limit should be 20 millisieverts. But in some scenario, we may expose to much higher radiation, such as accidental radiation exposure or military activities during nuclear war, or medical radiation exposure during radiotherapy, basically in the case of radiotherapy or surgeon. Radiation caused many significant drastic effect on the biological system, and radiation induced free radicals, oxidative stress, DNA damage, mitochondrial membrane potential, calcium level, and apoptosis. So therefore we need some drug which can help us to protect such in such case scenario. The first objective, evaluation of the radio protective efficacy of NET in neuro 2 cells. In methodology, cells seeded in 4 groups: first control, second radiation 20 gray, and
excuse me, Ravi sir, actually your slides is not visible. This is freezed.
Sorry sir, not visible.
Only show first slide. Yeah, now okay.
Now it's visible.
Yes.
Okay sir, sorry to interrupt. Uh, this slide is visible to all?
Yes, visible.
Okay, sir. In methodology section, we seeded cell in 4 groups: control radiation, net plus radiation, and net only. After all treatment, different study was conducted to evaluate the effect of net pretreatment on irradiated cells. Here we use cobalt 660 as a source and dose 20 gray. And different parameters we conducted: toxicity of net, neurotoxicity of ionizing radiation, DNA damage, cell death, mitochondrial health, ROS, and calcium level. Toxicity effect of net and ionizing radiation in neuro 2 cells was determined using MTT-assay. In figure A, it was found that net uh, has no significant neurotoxicity even at a higher concentration, 1 mg per ml. It's indicate NAT is a very safe drug to use. In figure B, it was found that LD50 was 20 gram in neuro 2 cells at 72 hours. Now, the radioprotective efficacy of NAT in neuro 2 cells was determined using MTT assay, and it was found that radiation significantly reduced approximately 50% cell survival, but NAT pretreatment 3 hours before radiation at a concentration of 0.04 microgram per ml. So maximum radio protection around 83% in net pretreated irradiated cells. To confirm this study, we conducted another set of experiment, radiation-induced cell death and its protection by net pretreatment in irradiated cells using PI exclusion method using flow cytometry. It was found that radiation significantly increased cell death, but net pretreatment helped to reduce it at a significant concentration, 0.04 microgram per ml. Again, we confirm this is study by another set of experiment. Here we use radiation-induced toxicity effect in neuro 2 cells by measuring the cloning formic unit. From this study, it is found that LD50 was 5 gray and LD90 was 6.5 gray in neuro 2 cells. In figure B, it was found that net pretreatment helped to enhance survival fraction in irradiated cells, and it was found that maximum around 80% cell survival in net pretreated irradiated cells. As we know, radiation significantly induced DNA damage, so we conduct DNA damage assay, and the role of net pretreatment in DNA protection in irradiated cells by measuring comet assay, and it was found that radiation significantly increased DNA damage. Uh, we can see in figure A, radiation significantly increased comet tail, which is a indication of DNA damage, and net pretreatment helped to reduce radiation-induced DNA damage. Second objective, evaluation of net pretreatment reduce oxidative stress and mitochondrial dysfunction in neuro 2 cells. For this, we conduct a number of experiments, and in figure A, it was found that radiation significantly increased ROS level, but net pretreatment helped to reduce ROS level in neuro 2 cells. This experiment conducted by DCFTA assay by flow cytometry. Now in figure B and C, we can see radiation significantly increased mitochondrial ROS level and mitochondrial content, but net pretreatment helped to reduce mitochondrial ROS level, but it maintained mitochondrial content. Again, the neutralizing effect of NAT on radiation-induced alteration in mitochondrial membrane potential in neuro 2 cells was analyzed by using rhodamine 123 fluorescent probe, and it was found that radiation significantly increased mitochondrial membrane potential at 48 hours and 72 hours, but NAT treatment helped to reduce mitochondrial activation, or we can say disturb the mitochondrial membrane potential by radiation, which is prevented by net pretreatment. Now, after conducting mitochondrial loss and mitochondrial membrane potential, we conduct a mitochondrial calcium level and cytoplasmic calcium level. In figure A, it was found that radiation significantly increased cytosolic calcium level, and net pretreatment helped to reduce it. But in case of 48 hours, it significantly increased. But in case of mitochondrial calcium and endoplasmic reticulum stress, in figure B and in figure C, it was found that radiation significantly disturbed mitochondrial calcium level and mitochondrial calcium level and endoplasmic reticulum calcium level, but net pretreatment helped to reduce mitochondrial calcium as well as endoplasmic reticulum stress. So from this, we can conclude net pretreatment help to reduce oxidative stress, mitochondrial membrane potential, and calcium level. Conclusion. The result of the present study indicated that net pretreatment to irradiated neuro-12 cell established mitochondrial redox hemostasis by inhibiting calcium migration from cytoplasm to the mitochondria. Net pretreatment to irradiated cells helps to protect the mitochondrial membrane potential and apoptotic pathway. So we can say NET may be used as a radio protection in neuro-2 cells because it may reduce oxidative stress, DNA damage, and maintain mitochondrial health as well as calcium level. This work is already published last month in OMCL Hindawi Journal, Impact 7.3. Thank you.
Thank you so much, sir. Nice presentation. And now the next participant is Alia Firdaus. Next participant is Miss Alia Firdaus.
Yes, sir. Is this visible?
Yes, visible.
Okay, so good afternoon all.
You are in YSP category?
Sorry?
Young Scientist poster?
Yes, sir.
You are in Young Scientist poster category, right?
Yes.
Okay, thank you.
Okay, so I'm a PhD scholar. I'm doing my PhD from Integral University, Lucknow, under the supervision of Dr. Uzma Faq. So this is my poster. The title of my poster is The Pesticidal Potential of Wheat Plants Against Some Common Phytopathogens. As we all know that agriculture pest management is a big concern nowadays and also from last many years. As compared to the other plant parasite, fungi causes the greatest impact or damage our crops more. So, besides this harmless and non-phytotoxic, it has been proved that plant extract exhibit a great and good effect on the phytopathogens and kills or inhibit. So, the plant-based pesticides are cheap, locally available, and non-toxic and easily biodegradable. Using biopesticides and biofertilizers together enhances the soil health and reduce the environmental pollution and also support the good and sustainable crop production. The use of application of agro-based chemical in the agriculture sector to protect the crop plants from invading and infecting pathogen can be greatly improved by employing the biopesticide or by the use of biopesticides in agriculture. Basically, the active principles of biopesticide vitamins